Okay, so we transition to a broad overview of sort of some more advanced topics in neurobiology and endocrinology and neuroendocrinology. And this is a completely obligatory step because what we've gotten through, thanks to the catch-up, thanks to BioCore, thanks to HUMBIO, whatever, is a whole bunch of basic facts about the workings of the nervous system and hormones. And of course, what happens at this point is you then find out, well, that's how it works most of the time. So what we're going to look at is some parallel dogmas that we all learn about the brain and hormones and version and seeing what's actually happening being far more complicated, far more interesting, far more allowing for informational subtlety being communicated by neurons, by hormones, all of that, and far more explanatory, most of all, about why you are different from the person sitting next to you. Okay, so the first piece of this, we bring in one of the gods of neuroscience, Dale. Dale, early in the last century, was the lawgiver. He was the person who went up to the mountains and came back with the commandments in stone with Dale's two laws of neuronal function. The two laws. The first one, which is when a neuron has an action potential, it releases the same neurotransmitter from each of its axon terminals, each of its 50,000 ones. Okay, so that was like an amazing observation. And people have learned that for centuries. And the sort of complication here, the qualifier, and one that's immensely interesting, is that sometime the most formal, strict version of Dale's law number one is incorrect in that sometimes you have neurons that from every axon terminal is releasing two types of neurotransmitters, the same two in each case, multiple neurotransmitters. And what we see here in this diagram, we see blue dots and orange dots. We see two different neurotransmitters coming out. Heresy, both of them in the same vesicle. In some cases, the multiple neurotransmitter scenario is you'll have a vesicle with the blue ones and a vesicle with the red ones. And here we've got the receptors for the two different types. Totally amazing. Is that totally amazing? Well, other than showing that you got to say Dale's law, saying whichever neurotransmitter or combination of neurotransmitters come out of this axon terminal is going to come out of all of them as well. Okay, so that's 21st century Dale. But you can see multiple neurotransmitters. What is that about? And what you see is they're typically two very different structural classes, chemical structures in neurotransmitters, ones that are made out of peptides versus ones that are not. If this is unfamiliar terrain to you, ignore this entirely, just structurally different. They tend to have different functions. They tend to work at different speeds, things of that sort. So that suddenly greatly expands one's neurotransmitter palette there if you could be releasing multiple messengers with different sorts of effects. One of them will interact with its receptor and change ionic events in a fraction of a second. The other one will change gene transcription over the next day or so. You just have a much, much richer palette of ways in which one neuron could talk to another. Now, the exact equivalent over in the endocrine system. What is it? This is not a Dale, but some equivalent sort of thing like, okay, the hypothalamus releases a hormone, which causes the pituitary to release its cognate hormone, CRH, corticotropin-releasing hormone, causes the release of ACTH, adrenocorticotropic hormone. This hypothalamic releasing hormone is the thing that tells the pituitary to release its hormone, which will then go to the adrenal glands and tell it to release glucocorticoids. So that's your basic cascade. The hypothalamus has one releasing hormone, which releases one of the pituitary hormones, the half dozen or so in the anterior pituitary. That's how it works. And in the early 80s, when people were first learning about CRH, when the structure of it was first solved, and that was the lab I was doing my postdoc in. People found something totally bizarre, which was there was more than one kind of CRH in the hypothalamus. There was more than one kind of hormone that could cause the pituitary to release ACTH. And it took people massive amounts to figure out that, like, CRH type 2 was actually the hormone oxytocin coming out of the hypothalamus. Vasopressin was another ACTH secretagogue, a thing that could cause the pituitary to secrete this. Epinephrine, norepinephrine, CRH, along with a whole bunch of these other hormones that collectively could release ACTH. Okay, my God, why the multiplicity here? And exactly the same theme as here. You begin to get control, different time courses, different mechanisms, all of that. What people began to see was for one type of stressor, the hypothalamus secreted CRH, oxytocin, and epinephrine. For a different type of stressor, CRH, vasopressin, and epinephrine for another, there was a coding at the level of the hypothalamus for what kind of stressor it was. Stressor, like you're psychologically stressed because you've been put into a new novel environment. Stressor, you've just had your limb broken. Stressor, massive, whatever, psychological, somatic, and it turned out there was coding of the inputs into the hypothalamus, so the cluster of oxytocin releasing neurons, the cluster of vasopressin, the cluster of CRH ones, all of that, and you got coding for that. Great, amazing specificity. That was so cool. People spent the whole decade sorting that one out. What's interesting about this is, when it's CRH plus this, you release ACTH, but you do something to blood pressure that's very different from when you have CRH with this. You do something to heart rate. You have coding for the additional effects. Yeah, release both of these, and they have different time courses, different mechanisms of action. Release this orchestration, stressor-specific orchestration, psychological stress versus physical, all of that. A temperature stress versus a blood pressure stress versus, and you get different coding. And somewhere in there, just to make life really complicated, there's also a corticotropin inhibiting hormone that plays. So this whole array, this whole orchestration, just to get more subtle control, because collectively they all release ACTH to different extents with different time courses and do additional things as well. So more specificity in that realm. Another way now in which poor Dale is going to go down the tubes, spatial distribution. What we just saw was neurons are way more fancy in terms of the messenger they release. They can have combinations. Endocrine systems remain more complicated. They may be regulating a hormone through multiple possible different messengers and combinations and all that sort of thing. Now we see something about spatial stuff in both the nervous system and the endocrine system. Nervous system. What is the dogma you were all taught, that we were all taught to say at birth, which is, okay, once a neuron gets sufficiently excited to have an action potential, the action potential functions in a, say it along with me, in an all or none manner. It's the same action potential jumping down the axon, and the same one at this first split into two branches, and the same one in the branching after that, and all of that. And it's the same action potential that propagates down to all of the 50,000 axon terminals. And here Dale comes back again with his second granitic law that we all bow down to, saying that when you get an action potential, the same action potential shoots down every single branch down there, and as we saw, releases the same neurotransmitter or the same combination of neurotransmitters. And his second law is the action potential is all or none, regenerates, and the exact same action potential that started here looks exactly the same whether you're here, here, or here. And of course, that one turns out to be more complicated. because it turns out that you could deposit different densities of ion channels, calcium channels, for example, at different branch points, and neurons do that. And what that does, you could do here, you could do it right down at the ends there, is you individuate, you fine tune, you accessorize, you make special the action potential coming down this branch versus that branch. And what you'll see is, depending on the density of some of these ion channels here at that, the shape of the action potential, the width of it, will vary. What's the consequence here? This is how long it's going on for, or the shape of the recovery period will change. The shape of the whole thing, depending on which branch it goes down, and the density of different types of channels in there. How do neurons regulate that? That is amazing, and is something we're probably going to look at later this week with emergence, all of that. How does it know to have this many of these channels in this one to get this kind of action potential versus this other many in this one to get this shape? Where's that regulation coming from? The main thing is Dale was wrong. It's not the exact same action, but the main thing is neurons have this informational control as to how strong, how long of a signal is coming down different branches. And there's even circumstances where a neuron appears to be able to decide nothing's going down this branch right now, to shut off that bifurcation point. So regulation on that level. Far more regulation than people ever anticipated. So Dale turning over in his grave for the second reason. Hooray, and action potential gets to all the axon terminals, maybe not even that all the time, but regulation along the way. Different branches are yelling their excitation in qualitatively different ways. The exact equivalent now with the endocrine system. Oh, very simple spatial stuff. The hypothalamus releases its hormone, CRH, for example, which goes to the pituitary and releases ACTH. Very simple. The pituitary is like the anterior pituitary, is this dizzyingly complex gland, and it releases like a whole bunch of hormones. It releases ACTH, which we were just looking at. Again, if this freaks you out, go to the catch-up section. It releases luteinizing hormone, follicle stimulating hormone, growth hormone, prolactin, thyroid stimulant, a whole array of them. And every single cell in the anterior pituitary does not have the capacity to release all these different, you got specialized cells in the pituitary. Corticotrophs release ACTH, gonadotrophs, somatotrophs, lactotrophs, thyrotrophs. Different cell types are making their characteristic hormone and secreting it into the bloodstream in response to the hypothalamus sending its characteristic hormone down to stimulate CRH, stimulating the corticotrophs to release ACTH, growth hormone releasing hormone, stimulating the somatotrophs to release growth hormone. Okay, so that's great. So we've got a whole bunch, at least 5 different cell types in the anterior pituitary. So the obvious spatial question now, do you have the somatotroph neighborhood? Do you have the thyrotroph neighborhood? Do you have specialized subparts? No, what you see instead is just a scattering of all the different subtypes. A very relevant question is to ask, what kind of neighborhood is this particular somatotroph cell, this particular thyrotroph cell, as shown here as follows. Here's one corticotroph cell that just happens to be in the neighborhood completely surrounded by gonadotrophs. In contrast, in this other part of the pituitary, here's one corticotroph cell surrounded by nothing but somatotrophs. And what you see is, for the same amount of CRH coming down, these two differ in how much ACTH they pump out. They differ in their sensitivity to the signal. Local control on that level. And what your handout has is the most god-awful diagram imaginable, and immediately ignore it. It's just showing with one of these types here, what are all the local signals coming from the surrounding cell types, because you will not only get a string of gonadotropes or strings, but a string of a mixture of them, all of that. What kind of signals are coming in there, modulating the sensitivity of it? Oh my God, incredible control there. Incredible control in that the same hypothalamic signal will have a different impact on pituitary hormone release depending on which part of the pituitary, what local little neighborhood you're tapping into. Okay, that's great. That's totally fascinating. But wait, aren't you just like dumping your hypothalamic hormone all over the anterior pituitary? So you're stimulating the really responsive corticotropes and you're also stimulating the less responsive ones. And what have people found since then? The work of a guy named John Porter showing, okay, so you got your hypothalamus and you got your pituitary, and it looks just like this, and you've got a vascular system, blood vessels coming down, called the hypothalamic pituitary portal system, carrying these hypothalamic hormones down to the pituitary. And what he showed is individual microscopic little threads of these capillaries have different concentrations of the same hypothalamic hormone. The hypothalamus is able to differentially sort of shunt a hormone more in the direction of this type of corticotroph or that type of corticotroph, that sort of thing. And you got control up on that level. And I spent 2 years of my life as a postdoc learning how to cannulate one single one of those blood vessels to look at it. And that was the most exciting thing since neuroanatomy. But what you see, a whole additional level of regulation here. So a neuron does not just send an action potential. It can finesse, it could massage, it could shape what sort of signal it is sending to different of the downstream neurons. A hypothalamus does not just send a secrete ACTH signal, it could control, is it sending it to more sensitive neighborhoods, the less responsive ones, and it's the same thing for every one of these. Good. Next thing where we see elaborations. And in terms of going through this, do not be memorizing all of this, just conceptually get a sense of what's going on, that you get heterogeneity of responsiveness, and the hypothalamus, you have heterogeneity of what sort of action potential is reaching the end, and you have control on this level and still poorly understood in both of these domains. Next thing to look at, checking the time, next thing to consider, negative feedback regulation. Okay, negative feedback regulation. You flush the toilet and you now refill the tank, and you need to have a negative feedback signal telling you when the tank is full. You need to have a way of measuring the amount of water filling up in the tank, a flotation device, and it needs to be coupled to something which, as it rises with rising water levels, puts a lid on top of the source of the water. You need to have a negative feedback signal there saying, ooh, I've gotten it into my head that I, the neuron, want to release this amount of neurotransmitter into the synapse, and it would be nice to know if I really did do that. Or I, this endocrine gland, wants to have this level of this hormone in the bloodstream. Is it here and thus keep secreting, or is it time to reach threshold and shut it off? Do you definitely shut it off if it's gone with it? You have to have negative feedback in these cases. And there turns out to be some very subtle, interesting ways in which that occurs in both the nervous system and endocrine system. Okay, back to our neuron before with its little crown here, and whether this is a neuron releasing one type of neurotransmitter or the weirdo specialized ones with two types, whatever, this neuron has gotten it into its head that it wants to release 11 units of neurotransmitter into the synapse, and it would like to know, did 11 units really get there? I need a feedback signal. And what you see is, sitting on the axon terminal is a receptor for the neurotransmitter you're releasing. That's crazy. The receptors are on this side, on the dendritic side of the next neuron in line. That's how they're talking. What are you doing having a receptor on the axon terminal for this neurotransmitter? This is the bookkeeper. As the neurotransmitter comes pouring out, 0.1% of it wanders around this way and binds to this receptor, which has to have a rule saying whatever number of molecules of neurotransmitter is binding here, multiply by 10 to the fourth, and that will tell you how much came out entirely. And if it's not at the level you want, start packing these vesicles with more neurotransmitter or less oil. You get a feedback signal. You have receptors that are measuring the thing that their cell secretes in a regulatory way. Term in the business, an autoreceptor. An autoreceptor, making perfect sense there, a presynaptic autoreceptor, a way of doing bookkeeping. And this is very logical, very straightforward. Things get more complicated when we have our scenario of the same neuron releasing two types of neurotransmitters from the same vesicle, because now you want to get feedback information on both, because almost certainly you are not just interested in how much of A you're pumping out and how much of B, but the ratio of the two. So you've got to have two types of autoreceptors here that could integrate the information that they're getting, turning it into a signal of not just more of this one or less of that, but do whatever to correct the ratio of the two. So that's happening. That's totally cool in terms of complicated negative feedback stuff. Then an additional type of negative feedback subtlety going on in the nervous system. So what did we learn from Dale's great-great-grandparents in terms of action potential? Neurotransmitter comes out from here, floats across the synapse, does something to this neuron, and something changes in this neuron as a result of binding to these receptors, and hooray, this neuron has just talked to this neuron. This is where the neurotransmitters are. This is where the receptors are. Information flows in this direction. And the complication came in as people began to learn there's a whole bunch of weirdo neurotransmitters that are made in the dendritic spine in totally the wrong place. And they're released not in response to an action potential, because you don't get action potentials at the dendritic end. It's released at various points and they travel backwards. They go in the wrong direction across the synapse. They are, quote, retrograde neurotransmitters. And what they're often doing is serving a negative feedback signal on, okay, we're very, very excited here. You may want to tone things down a little bit. Or we're doing fine here. If you want to dump more stuff on it, go. It's a feedback loop this way with these bizarre, unconventional neurotransmitters that are made in the wrong place and go in the wrong direction. As it turns out, chemically, structurally, they're often quite bizarre. And here's an example of how that system works, which is so elegant. You can't believe it. Okay, I assume most people in here at least are familiar with ATP. ATP is the energy currency of the cell. If that's just saturated you, that's plenty enough. ATP is the molecule that's the source of energy for cells. So we have two circumstances here. Suppose this is a cell that is well fed and has had so much nutrients, it is full of ATP. It's got so much ATP that some of it dribbles at the dendritic spine and goes floating backward. And bizarre as hell, nobody believed this at first, there's ATP receptors. And what is this neuron saying to this neuron? I have so much energy that I can afford to drool out some of my ATP. We're doing great here. If you've got something very, very, very excitatory to say to us, go for it. Dump as much as you want because we've got things under control here. In contrast, suppose this is a neuron that's running into trouble energetically. It's running out of energy because it's had a stroke or a seizure is using up its energy at a crazy high rate. It's running out of energy. Do not freak out at this biochemistry. If this is new to you, this is not pertinent. So what happens when you're running through your energy storage? Your ATP is broken down to ADP, which is broken down to AMP, which eventually is broken down to adenosine. Adenosine. If you have a neuron that's got lots of adenosine and not much ATP, this is a neuron that's having a major energy crisis. So what happens now, as adenosine builds up, it goes backwards as a retrograde neurotransmitter and binds to adenosine receptors here. And what is it saying? We're exhausted here. We can't do anything. I don't care how exciting your news is. Stop sending us neurotransmitter. Decrease the release of all. We've got a feedback loop here by which the energetic state of this neuron can modulate how loudly this neuron is yelling at it. And that's totally beautiful. This is the neurobiology of why you should not take exams when you haven't eaten for 3 days. Here's the neurobiology of why neuronal communication is sensitive to neuronal energetics, to energy availability. Very interestingly, in most individuals who have a grand mal epileptic seizure, neurons are firing like crazy, so that ATP gets broken down, and it is the adenosine building up in this exhausted neuron, floating backwards, telling stop releasing neurotransmitter. It's this buildup of adenosine which typically plays the role in finally stopping the seizure and terminating it. Okay, so beautiful negative feedback regulation. Neurons measure their own neurotransmitter release rates by having receptors of their own right here. And then we've got all these weirdo retrograde neurotransmitters that communicate modulatory stuff. This is a way for this neuron to tell this neuron how things are going here. And sometimes the message is, we're great, yell as loud as you want. And sometimes it's saying, if there's a way you could tone it down, we're falling on our faces here. So beautifully elegant regulation there in ways nobody ever anticipated. Now, negative feedback regulation complications going on in endocrine systems. Here's where we always get back to the toilet bowl model. You've got a threshold. You know how high you want the tank to fill up with water, and you have to have a means of measuring how much water there is in the tank, your flotation device. And as it reaches threshold, it's got to be set up that the flotation device being here shuts off the water spout, and you've got a negative feedback system. And it works wonderfully, and that's great. And here we see a perfect example of this. You are the hypothalamus, and you've decided you want to start a whole stress response. So you secrete CRH so that these guys secrete ACTH, so that the adrenals secrete glucocorticoids. And you want to know how much glucocorticoids there are in the bloodstream, because for this type of stressor, you've gotten into your head, we want this much glucocorticoids, whereas for this much milder stressor, you want this much. You've got to have a threshold, and you've got to have a means of measuring glucocorticoid levels in the bloodstream and have a negative feedback signal. And what you see is throughout much of the brain, you see totally straightforward. The more glucocorticoids you pump into the bloodstream of an individual, the louder of a negative feedback signal, the more you're pushing up the levels of the water in the tank. And the higher the levels, the more you shut off subsequent CRH release. Just negative feedback. And then in the 60s, endocrinologist named Mary Dahlman discovered there's an additional type of negative feedback going on. And this is right in the hypothalamus. And there, the brain is not caring about how much glucocorticoids there are in the bloodstream. It cares about how fast they have been rising. Rather than paying attention to the level, it's paying attention to the rate. So that when it's rapidly increasing, you tone down CRH, and that's occurring in the first 2 minutes of a stress response just to fine-tune stuff. And then 2 minutes in or so, you switch over to level sensitive feedback. And it's just how much is there? And the more there is, the closer we are to threshold, and it's time to shut this off. These are the same neurons that some of the time they're measuring how much glucocorticoid thermal in the bloodstream. Some of the time they're measuring how much has it changed in the last minute. This produces two totally amazing sort of challenges here. The first off is, how do you code for this? How do you code this? Sometimes this cell, this neuron that's a source of the feedback signal, it cares about how much versus some of the times rate of change. What's the nuts and bolts of that? And is it being so literal about the rate of change that as far as this part of the brain is concerned, going from 1 unit of glucocorticoids to 2 in the next minute is exactly the same as going from 101 to 102. Huge, complicated feature in terms of what the wiring is for that. Not fully understood yet, but fine feedback some of the time very early in a stress response. You're not so concerned with the levels. You just want to know that it's rising fast enough. And once you get up towards plateau, then you're caring about the levels, the switch back and forth. A very cool thing about that study, by this person, Mary Dolman, who was the first fully tenured neuro professor woman at UCSF, was chair of the department there for years. Wonderful person who was a mentor and friend who died during the pandemic. But here was the coolest thing of all, which is when she first published about level sensitive and rate sensitive negative feedback, it wasn't from an experiment. It was a theoretical paper. Her origins were in math, and she did this whole informational control analysis and saying, just from what we know about glucocorticoids and what happens under it, there's got to be two different types of feedback. There's got to be this domain that does the normal familiar hormone stuff, but also to explain these findings, there's got to be a domain where for the first couple of minutes it's concerned with rate, and like, who knows, but this is what my model suggests. And then everybody on Earth went out, including her, and showing this is exactly what was going on there. This was one of the most beautiful examples of modeling simulation, and this was back when computers were the size of Rhode Island. This was her just sitting with a pad of paper and a pen and a abacus or something, figuring out it's gotta work like this. This is the only way to explain what I'm seeing in my experiments, and that turning out to be exactly the case. Tell you what, before we get into the next additional complication, let's call it a day at this point, and we will pick up with how both neurons and endocrine cells change the number of their receptors under different circumstances. They will change the workings of the receptors under certain circumstances. And getting to our most important concept in here, some of the time a neurotransmitter or a hormone is not having an effect. What it is doing is changing the ability of some other neurotransmitter or hormone to have an effect. Rather than a causal role, a modulatory role. And just to give dramatic foreshadowing as to the concept that will get pulled into here, sometimes we're saying, what's the effect of this neurotransmitter or hormone? Well, it depends. It depends on whether this other one is present or not. We have a neurotransmitter interaction or hormone-hormone interaction, and you know what that looks like.